Okay, on the sound of the chime. Thank you all for coming this morning for the first talk in the Phylogenetic Comparative Method sessions. So I'm Ben Auerbach, I'm the first speaker in the session. I'd also like to acknowledge my co-author who's not here, Liz Augusto, who is at Indiana University School of Medicine, who is a very adept studier of evolutionary comparative methods. For those of you who are unfamiliar with my work, I am generally a primate person. I tend to study primate morphological variation. So this is a little bit a field, but just to put things in perspective, these are data that I collected over 20 years ago, and thank you, pandemic, when you pull things out of mothballs and you're like, I looked at this 20 years ago and I wasn't satisfied with the method, so I never published on this. And now the method's caught up with my questions. So we're talking about ungual phalanges. So for those of you unfamiliar with these are the claw-bearing bones at the tips of the digital rays that you find within most vertebrates. And if you look across mammalian species, you can kind of get an idea that there's a lot of different functional uses for claw morphology. So we can kind of typify these by shape. These are images that I lifted from McLeod and Rose's paper in 1993, where they used eigenshape analysis to compare the shapes of these. And basically what they show is that you can generally correlate the shapes of these bones with the functionality of the organisms that are using them. You can kind of put them into these kind of broad locomotor groups. I think most people are probably familiar who do mammalian research with these kind of locomotor guilds. Now, of course, there's a lot of controversy ecologically about how you define species based on locomotion. But notable papers that have worked on this particular question, looking at one particular element, granted locomotion is the entire skeleton, so looking at one element is a little bit reductionistic, but still, when we're dealing with fossils, and that's why they were interested in this in the first place, sometimes you end up with ungual phalanges, and it's useful to try to typify locomotion based on whatever evidence that you have in the paleobiology. So Blair Valkenburg was probably the person who really made this an area of study. She basically showed that you can find locomotor signal in the shapes of these bones. This was followed in 1993 by the McLeod and Rose study, where basically they looked at the overall shape of the bones and said, yes, the shape of these bones does roughly correspond to these locomotor groups. They looked at 5 locomotor guilds. And then there's been a series of other papers since then. And of course, the most recent one that I want to point out is the one in the lower right-hand corner. This is from 2020, where they looked at biomechanical signals. And they weren't really putting it into an evolutionary framework, but they also kind of used a broader set of locomotor groups to kind of say, okay, well, the shapes of these bones seem to correspond with what these organisms are doing. The problem that I've had with this literature is that while it is useful, it is largely descriptive and pattern-based. Not to say what we do with phylogenetic comparative methods isn't also kind of pattern-based, but we at least have informed models, right? So we have ways of saying what model seems to best fit the data that we have. So without further ado, this leads me to kind of the questions that I was interested in when I pursued this study. So the first one was asking, are there phylogenetic models? If we use phylogenetic comparative methods, can we model different evolutionary regimes potentially that are taking place amongst these particular bones? So for example, does your relatedness to other taxa say something about the shape of your bones more than what you're doing with the bones? So I asked the questions, okay, if you're a mammal, are you a marsupial versus a placental, right? Is that maybe one separation that we see? It's a very rough separation, right? Or more specifically, if we look at kind of order level, morphological variation, does that better explain what we see in terms of selection than, say, what's going on with locomotor groups? And the other question I had is more of a biomechanics question that's informed by this, which is to say, well, if we look at the biomechanics, do the biomechanics actually say something about locomotion as well? Because a lot of these studies have looked at shape, but they haven't actually looked at, say, bending rigidity or resistance to torsion. And I think that that is a little more meaningful possibly than shape. So shape may be telling you something about phylogeny, and the biomechanics may or may not be telling us something about locomotion. So that's really the two sets of my questions. So the sample, which you cannot read, is 42 different taxa. These are the exact same taxa that were used in all those classic studies. So I thought, okay, if I'm going to do this, I want to go after the exact same sample that was used previously so I can test their particular questions using the exact same sample. In most cases, the exact same individuals, because as it turns out, these bones are typically lost in collections, because when they go to skin mammals, the unguals tend to stick around with the claws, so they don't tend to stick around with the rest of the skeleton. I took a bunch of linear dimensions off of them. There's an example here of what these bones look like based on 5 or 6 taxa that we see here. I made calculations from those linear dimensions. So the ones that I'm going to be focusing on are basically the middle of the phalanx, its cross-sectional shape, because that tells you how tall and wide it is. And of course, the wider it is, in theory, wider relative to tall phalanges are associated with organisms that tend to dig, while more narrow ones are associated with organisms that tend to either be generalists, or they tend to do things like climbing, or they tend to do more grasping. So we do tend to see these types of behaviors. Of course, I'm putting these into these locomotor guilds. So the locomotor guilds I'm looking at are arboreal and scansorial, cursorial, fossorial, and generalist. So 5 locomotor groups. I also calculated cross-sectional geometric properties using a PQCT scanner. So I put these little bones into the PQCT and got cross-sectional shapes, so I was able to get the actual biomechanical properties out of them. So to do my analyses, since this is a phylogenetic session, just so everyone knows, I used Bayou and Aui together. So basically, I used Bayou to create my models and then ported those over into Aui to run the analyses. I subjected them to all the different types of OU models that are in with Aui, so ones that let you vary alphas, for example, versus not varying alpha. And then also subject them to Brownian motion, just because we always do that. And then I also did comparisons between them using robust Bayesian ANOVAs to compare the cross-sectional properties to look at the locomotion. So basically, these are the kind of the two ways I went about doing this. And I'm happy to talk more about the methods, but we need to get to the results. So if anyone has questions about methods, find me after. So this was the surprise. Locomotion is a very bad explainer. At least the 5 locomotor categories I'm using here is a very bad explainer using phylogenetic comparative methods, right? So all my OU models basically said, hey, look, who you're related to better explains selective regimes than what you're doing with these bones. And this kind of completely runs against what almost everything in the literature suggests about these. So I'm still a little skeptical about the results, but if anyone has ideas or if anyone would like to talk about this, I am very open to hearing your thoughts, because this really completely contradicted what I thought was going to be the case. And just to kind of give you all an idea, here are 2 of the dimensions. So on the left is that ratio of the height and the width of these ungual phalanges. That's cross-sectional shape. Again, the flatter the bone is, in theory, you're fossorial, the taller the bone and the more narrow the bone is. Think about like a cat, for example, right? Long and thin and narrow claws. You would expect them to be more grasping or more kind of using them for holding onto, say, exactly a substrate, for example, when they're climbing up it. As it turns out, the locomotor groups don't fit the models at all. Instead, what we're seeing here is something that's more related to who you're related to. When it comes to J, which is torsional resistance, so the ability of these to resist torsion over here on the right, there's actually almost no selective regime changes that are showing up on here, if you notice. There's a handful that are showing up in odd taxa like bears and also on agoutis. But most of these organisms are not showing that pattern. So this makes me maybe a little skeptical of what we're seeing. Just to kind of throw some taxa on here so you can see what we're actually talking about. You can actually see that some of these selective regimes actually just match which order you're in. So the difference between carnivora and rodentia seems to be what's driving the difference. And actually, we'll take a closer look at carnivora in a minute. But there's a pattern here that definitely seems to not be having anything necessarily to do with what you're using these for, because, just to make a point, we've got a lot of locomotor groups represented within the same selective regime. And that, to me, says, okay, either our categories are too broad and they're not getting at the specifics of what these organisms are doing, but even if they were, we know that these organisms are not doing the exact same types of functions with their manus. So it doesn't make a lot of sense that we would use that as an explainer. Okay, so the results are basically that no, models based on taxonomic order level or family level evolution best fit the data for cross-sectional shape resistance to torsion. So it's not based on locomotion. It's based on who you're related to. Okay, and just to drive this point home, here are the differences between our locomotor groups in two of these dimensions. Again, this is the relative shape. I looked at more dimensions than these, but this is a talk, so you can only go through so many dimensions. But this is that kind of cross-sectional shape, and this is the actual biomechanical resistance. You can see that they all overlap in biomechanics, basically. They all have the same torsional rigidity. And when you're looking over here on the left, you can see some pattern differences, but statistically they're non-different from each other. If I look at the Bayesian ANOVA that I did, so these are my Bayesian ANOVAs comparing between my 5 large kind of order-ish level, we know marsupials are not orders, but like order-ish level taxonomic groups, we can basically see that the differences seem to be driven by, for example, here in the cross-sectional shape by carnivores versus anarthrons. Now, I should caveat to say I only have 4 xenarthra here, 3 armadillos and 1 sloth, so we don't have a great diversity in that. There are a lot of carnivores in this sample. And if we look at, for example, J, that torsional resistance, we can see that it's, again, being driven by differences between carnivores and xenarthrans, but also rodents seem to be playing a role in this too. So those orders, there's a lot of speciose diversity showing up in those orders. It seems to be relating to these selective regimes. So the second part of my results is basically that does the cross-sectional morphology and the biomechanics of that cross-sectional morphology tell us anything about what's going on in terms of locomotion? And the answer to that question is unfortunately no. And again, not really matching with what the literature has said, but then again, most people have not done biomechanics on these. They've looked at the shape, but they haven't looked at the actual bending rigidity in these bones. So in conclusion, here are a few take-homes. So first, there's no statistical difference in resistance to torsion or bending resistance among the locomotor groups. So we need to probably do a little bit more actual biomechanical study of these bones before we just rely on shape to tell us something about the differences among them. There is an important subphylogenetic signal in distinguishing the shape of the ungual phalanges among mammals that is not ignorable. They reflect shared selection regimes among families or orders more than locomotor or functional related use. And to that point, let's take a look at the cross-sectional properties in the shape. Yeah, 2 minutes. Okay, so here is that result, a phylogenetic result, and we're going to just focus in on the carnivores, and this just kind of drives the point home. So if you're looking at the carnivores here, you're seeing a whole bunch of different adaptive regimes that are showing up within carnivores, but at the same time, they're all doing very different activities with their manus. And to me, that basically says that there is a lot of relatedness signal here. So there is some selective event that's taking place fairly early when you see maybe family-level speciation. But beyond that, if they're specializing in terms of their activities, the actual shape of the unguals is not evolving to match what they're doing with them. So this tells us that there's a disjunct that's taking place between these. Okay, so final conclusions. One thing, caveats, it's very difficult to disentangle function from phylogeny. I think we all know that this is a problem. So closely related taxa may be sharing very broad locomotor function more than distantly related taxa, and so this is one of the problems, but this is why we have phylogenetic comparative methods, but it can also sometimes be difficult if your groups are too coarse. So there's not a lot of data here, right? 42 taxa, not a ton of data for a phylogenetic comparative methods study. And also 5 locomotor groups are probably not fine-grained enough to tell us something. So we probably might test this against other models. They aren't correlated in my data, but still it's a small data set. And also, I think it's important to say if you're going to make taxonomic comparisons among groups that you should probably take phylogeny into account, which is, of course, always the message. All right. Thank you all. I want to thank the Smithsonian for letting me scan their bones, Brian O'Mara, who was useful in giving me some advice in doing this, and Ken Rose and Chris Ruff, both long since retired, but both of them who provided early support for this. Just a quick note, I'm plugging my student, Kelsey O'Neill. Her poster is tomorrow on primates and covariation in the skeleton and evolutionary covariation. Information's on the screen, and you should go see it. Thank you all very much.
Okay, well, I will start anyways. Hi, everybody. I am Corinthia Black, and I'm a postdoc at the National Museum of Natural History. And today I'm going to be telling you about some of the work that I have been doing during my postdoc in the Wood and Schultz labs, kind of looking at how this extended phenotype is affecting the somatic phenotype in spiders. So when we think about evolution of phenotypes, we generally focus on how phenotypes are changing within a group of organisms. And we tend to really overlook how those changes influence and alter the environment in which the organism lives, and how it kind of creates this feedback loop where these environmental changes tend to lead to further changes of the somatic phenotype of the organism. Excuse me, I'm still slightly recovering from COVID. So what is the extended phenotype? Well, this is a concept that attempts to describe how, when, and why the genes of the organism alter the outside world. There's some really classic examples of the extended phenotype. We can see these in social interactions, like these dominance displays between deer, the construction of structures, which the beaver's dam is a really important one, kind of completely changing the entire environmental system where the beaver has built its dam. And then also these host-parasite interactions, where we can manipulate others, like the zombie ant fungus, where it's taking over this ant entirely in order to kind of feed itself. So these types of interactions can be seen across the animal kingdom. And the web in spiders is another classic example of this extended phenotype. This web modifies the spider's environment in many different ways. It has helped to expand foraging opportunities, as well as enhance the spider's ability to restrain, detect, and also intercept prey. And although all spiders create silk, and they use them for various reasons, like protecting egg cases, forming retreats, that kind of stuff, not all spiders actually use silk in order to capture prey. So actually about 50% of the 52,000 species of spiders are free hunting spiders. They're running around catching things, whereas the other 50% are using webs in order to capture prey. And this free hunting has actually evolved many, 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 many different times across the phylogeny of spiders. So there have been some other studies that have attempted to examine how this extended phenotype has influenced the evolution of somatic traits in spiders, but there's been some conflicting evidence. So, for example, studies that have focused on general morphology have shown that the web has little to no effect on the spider's phenotype, kind of suggesting that this extended phenotype does not really promote evolutionary change. But in studies where functionally relevant traits have been explored, those mechanical properties of the web seem to replace certain aspects of the spider's internal neural processing, especially like vision and sensory perception. And over here on, I think your right, I always get those mixed up. So on your right, you can see on top here, we have a spider with really tiny eyes. This is a web builder. The neural processing system is kind of small too. The one on the bottom has these huge eyes and this large internal processing as well, and they are hunting spiders. So for this study, we wanted to know if functionally relevant traits that are directly involved in prey capture were influenced by web building. So specifically, we hypothesized that the web, as the spider's extended phenotype, can functionally replace strong bite forces because the web is acting as this extension in order to immobilize the prey for the spider. So this would mean that our free hunting spiders would have stronger bite forces than our web building spiders. But how a spider captures its prey is actually quite complicated. So it was way more complicated than we imagined. We do know that the chelicerae, they function as two different joints. There's a joint between the fang and the cheliceral base. This is a really simple joint, movements on a single plane. There's a nice pivot point. We can model this really well. But the joint between the cheliceral base and the carapace is incredibly complicated. It's actually controlled by 8 to 9 pairs of muscles. There's no direct pivot point that we can find, but there is one pair of muscles that seems to control unified movement of the two cheliceral bases, suggesting that this centrally located muscle is critically important to the function of bite forces in spiders. And that muscle is called the intercheliceral muscle, and it's shown in light blue here. It attaches to the intercheliceral sclerite, which is a tiny little sclerite that sits between the two cheliceral bases. This is held in place by connective tissues, so any force that's applied to that sclerite by the muscle results in simultaneous movement of the cheliceral bases during prey capture. So we wanted to test if the forces applied by this intercheliceral muscle were weaker in the web-building spiders than they were in the free-hunting spiders. So to do this, we took CT scans across the phylogeny. So we ended up getting muscle forces. We modeled muscle forces from these preserved specimens. We tried to get a good representation of web building versus free hunting across this phylogeny. And in this figure here, each of these arrows represents 55 of the specimens that we got muscle modeling factors for. So in order to calculate the amount of isometric contractile force, we developed a method that helps us to estimate the force vectors and the number of muscle fibers from preserved specimens. So we started by taking our CT scans and aligning them so the attachment point of the legs to the body were parallel in the XY plane. We then fitted a polygon to the intercheliceral muscle as close as we could to the carapace as possible while retaining the same Z coordinates. So on the left-hand side here, you can see a lateral cross-section with that polygon in red. And then on the other side, you can see kind of a top-down view where we've kind of cut through the carapace right where that polygon is located on that intercheliceral muscle. So within the polygon, we recorded the number of fibers from the longest and the widest point of the muscle. We then created a grid with evenly spaced points in the R package, StatSpat-geom, which is a fun package to say. We then fitted the polygon coordinates to this grid, and we retained the coordinates that fell within the grid and used those to calculate our isometric forces. So these points represent the estimated muscle fibers and their placement within the polygon. And when we compare those to actual counts in the spiders, they're almost exactly the same. There's a little bit of variation, but not very much. So once we had our coordinates, we calculated the force vector of each of the muscle fibers, which are represented by these little small arrows. And then we summed those vectors to produce a single isometric contractile force value. And once we had all of our force values, we size corrected those so we could compare across our spider species. So to test these hypotheses, that the web-building spider was going to have weaker forces than the free-hunting spiders, we performed phylogenetic generalized least squares to determine if the presence of the web correlated with weaker forces while accounting for those phylogenetic relationships. And what we ended up finding is that there is a correlation where we do see web building seems to correlate with our weaker forces. So in this figure, we have reconstructed the ancestral states onto the most recent and robust phylogeny, which was by Kalal and others in 2018. We have reconstructed the ancestral states for the total isometric forces for the intercheliceral muscle. And the lighter colors on this figure are representing weaker forces, whereas the darker colors are representing stronger forces. The circles at the tips of the phylogeny represent if the species is a web builder in white or if they are a free hunter in black. And when I highlight just the web-building species, we can see that the majority of these have weaker forces. So our little white bubbles are kind of matching up with the white tips of our phylogeny. So this supports that the web builders do have weaker bite forces, as the web's kind of acting as this extension to immobilize the prey. However, when we look at the free hunting spiders, we didn't see overall that the free hunting spiders had stronger bite forces. In fact, we see quite a bit of disparity in bite forces of the free hunters. It's about 2.5 times more disparate than they are in the web-building spiders. So this was really unexpected for us, but it was kind of a cool thing that we found. And it really suggests that these free hunting spiders have evolved many different prey capture mechanisms, and that these behaviors to immobilize prey really should be treated as a continuous trait rather than this binary, whether or not they're using this web versus if they're not using a web. For example, with the free hunting spiders, there's really a variety of ways that they can immobilize prey. Some spiders, like the wolf spiders, actually have these spines on their legs that they use to kind of catch prey and hold it in place so they can bite it and envenomate it. Whereas there are other spiders that rely 100% on their chelicerae in order to bite and hold that specimen in place. And there's pretty much everything in between. So in conclusion, we did find some evidence that the web itself is acting as this extended phenotype for the spider. It is influencing the function of the jaws by reducing bite forces within our web-building spiders. But we did find that free hunting in our spiders really should be treated as a continuous trait, which we should really apply to our web-building spiders as well, in order to kind of more understand how the function is changing across the spiders. And something that I didn't really talk about, we did look at the shape of that intercliceral sclerite, and we did not find any sort of correlation at all between web building and free hunting. So what this really kind of suggested to us was that these functionally relevant traits are really the best to test these kind of questions of how the extended phenotype is really influencing the somatic traits within the organism. Of course, this is a ton of work. A lot of people helped out with getting scans, segmenting things, and of course, we have to recognize our funding sources through NSF. And with that, I think I have like maybe a minute for questions. Sure.
Really cool talk, thanks. I was curious about the sort of like exception to the rule of the weak bite force in the web building spiders. Do you have any sense of like who is this spider that has strong bite force and
Yeah.
why?
So we did look at that. There were 3 species that had stronger bite forces in the web builders. One, we don't really know anything about its behavior. The other 2, we know that they do not have sticky silk. So they're kind of more using these webs as a means to feel if something's there. And they have to run out and catch that organism that enters into their web. It's actually really cool, and I'd love to talk to you more about it, but I think that sufficiently answers the question. Yep.
Given that you had to standardize the position of your scans, it occurs to me that the direction of those vectors might also be biologically relevant. Have you considered looking at that to see whether there's differences in those as well?
We did. That is something that we did include. We didn't find a lot in the direction other than it seems like that kind of direction's evolving under an OU model. So there seems to be like a restriction in the direction of which that muscle is pulling. Yep.
I was wondering if you explored the association of bite force with body size and type of prey that the spider ate.
Yeah, we did not look at the type of prey. So we got our force values, we corrected it by size. We didn't really look too much at how size was affecting things, but I can talk to you more about it. I don't want to take up other people's time. I think I'm good to go, right?
So I'm Mariam. I'm a first-year master's student at McMaster University, and I'm here to kind of present to you some work that we're trying to finish up and hopefully write up, and then kind of introduce the future direction of my research in my current lab. And so sexual dimorphism is a really common trait observed across the animal kingdom. And we often discuss these traits in terms of their function in inter and intrasexual competition. But a really important feature of these traits is that they're actually incredibly nutritionally plastic, meaning that individuals that develop under poor nutritional conditions, or rather under really high nutritional conditions, can evolve these really enlarged, exaggerated traits, whereas individuals who are reared under some form of genetic or environmental stress or poor nutritional conditions will not. And a really classic example of this high nutritional plasticity for a sexually dimorphic trait is in the case of the narid fly, which these species exhibit male-biased sexual size and shape dimorphism. But when reared under poor larval nutritional diets, they will actually develop to be nearly monomorphic. And so sexually dimorphic traits tend to be under strong directional selection, directional sexual selection. And so eventually what occurs is that these traits will rise to fixation in a population such that we're depleting the genetic variation in these traits. And while this is what you would expect, in natural populations, we actually see high genetic variation for sexually selected dimorphic traits. And so this is referred to as the Leck paradox. And the genic capture model is one model to sort of resolve this Leck paradox and explain why we see such high genetic variation in sexually selected traits. And essentially, it's proposing that sexually selected traits can co-opt variation and condition such that they're still maintaining this heritable genetic variation. And that's what we're seeing in that narid fly example that I brought up earlier, where each point here represents a trait that is under increasing intensity of sexual selection. And so as traits become more sexually dimorphic, we see them also become more condition dependent. And so if sexually selected traits can co-opt genetic variation and condition, thereby generating heritable genetic variation in the dimorphic trait, then we expect sexual dimorphism and condition dependence to co-evolve. And within species, we see what I just showed you, where as the intensity of sexual selection increases and these traits become more dimorphic, we also see heightened condition dependence for these traits. And then across species, what we'd expect to see is that species that exhibit heightened sexual dimorphism for a given trait should also evolve this heightened condition dependence for that trait. And so to test the predictions of the genic capture model, we looked at the macro and microevolutionary patterns of sexual dimorphism and condition dependence across the Drosophila melanogaster species group. And not surprisingly, this species group contains our model species, Drosophila melanogaster. And we see a lot of variation in the extent and direction of sexual dimorphism across our phylogeny. And then in left, you will see one side of the spectrum, Drosophila prolongata, which exhibits this really strong male-biased body size dimorphism. And it also, they also express this really classic exaggerated male-biased trait in the form of those enlarged patterned forelegs you're seeing in the males, but not really the females. And then on the other side of the spectrum, I have Drosophila melanogaster, which exhibits this female-biased body size and leg dimorphism that is common across most of the phylogeny, as you will see. And so we also see variation in the intensity of sexual selection acting within this phylogeny. And we see a lot of intrasexual selection in the form of male-to-male combat, where males will use their forelegs to box or fight with other males for access to female mates. And then we also see a lot of mating behaviors, mostly focused on the wings, where we call it wing displays, where the males will display their wings to kind of reluctant females. And we also see some kind of, in some species, we see these like leg displays that are similar to the wings. And so we manipulated condition by generating these fully fed, high-condition flies that were allowed to be, that were reared on food up until adulthood. And then we also generated food-deprived, low-condition flies that were removed from their food source near the end of the developmental period. And so we measured thorax length as an estimate or a proxy of body size. Then we measured foretibia length and width and tarsus length to kind of get at the sexual selection that we believe these traits are under. And we also measured wing area. Again, this is related to their use in that mating behavior. And then we use these estimates to measure, or we use measurements to estimate sexual dimorphism and condition dependence in our species. And so we used ancestral state reconstruction to model the evolution of sexual dimorphism and condition dependence across our phylogeny. And on your left is sexual dimorphism in mean tibia length for each species computed in high condition individuals. And as you can see, most of our species exhibit either monomorphism or weak female bias dimorphism in tibia length, with the exception of that one species I had brought up earlier, Drosophila prolongata, who exhibits this unique male bias dimorphism in sexual dimorphism in tibia length. And so, under the predictions of the gene capture model, we would expect that just as we see this really heightened male bias dimorphism, we also should expect heightened condition dependence. But that's not what we're seeing. When we're looking at the phylogeny for condition dependence in males for tibia length, we see this moderate condition dependent that is quite common of the rest of the phylogeny in that species. And so when we look at a trait that we expect to be under less intense sexual selection, in this case, wing size, again, Drosophila prolongata will not as severe as in tibia length. We're still seeing this unique male bias size dimorphism. And then when we look at the condition dependence again, we're seeing moderate condition dependence that is common across the phylogeny. And so we use phylogenetic independent contrast to model the evolution of sexual dimorphism and condition dependence across the phylogeny while accounting for the phylogenetic relationship between species, assuming a Brownian motion model of trait evolution. And if you look at the top left, I've displayed sexual dimorphism in high condition versus low condition individuals. And our points don't really deviate from our one-to-one reference line, suggesting that sexual dimorphism isn't really changing across our treatment conditions. And then similarly, when we look at condition dependence in females versus in males, while we see more variation from our one-to-one reference line again, condition dependence is pretty common across the sexes. And to really get at the question of whether or not sexual dimorphism and condition dependence are co-evolving across our phylogeny, we're looking at sexual dimorphism in high condition individuals versus condition dependence in males, and we see a very weak correlation across for these two traits. And one point that I really want to bring your focus to is that this contrast that's showing this really strong sexual dimorphism is only weakly condition dependent. And so these phylogenetic independent contrasts that I've showed you, we're only looking at tibia length. So to see if we're still seeing the same pattern across our traits, I've plotted the correlation in sexual dimorphism and condition dependence across the 5 traits that I mentioned earlier. And displayed here is an example in Drosophila melanogaster. And so as you can see, we're seeing this strong positive correlation across both the sexes, where our most sexually dimorphic trait, which is in the far, which will be on your right, wing area, is also very strongly condition dependent. And to sort of bring it back to that classic example of what we would expect under the gene capture model, again, and keep in mind that this is referring to this male bias dimorphism. So as male bias dimorphism increases, we also see that subsequent increase in condition dependence. And we're seeing this relationship in males, but we're not seeing we don't have any strong predictions about how this would relate in females. And so because we are looking at a phylogeny that exhibits both female and male bias dimorphism, we've computed our measure of sexual dimorphism a little differently. And so an equivalent line to what you're seeing in that Bonduriansky 2007 paper is shown in that bottom quadrant, whereas male bias dimorphism increases, we're seeing an increase in condition dependence. And we would expect to see that reflected in a positive, negative, strong negative correlation between sexual dimorphism and condition dependence. And again, we expect this in males, but we don't really have any strong predictions about the correlation that we would expect in females. And so when we look across, we look at the correlation between sexual dimorphism and condition dependence across our traits for the entire phylogeny, we're seeing, for the most part, the strong positive correlation that is in males as well as in females. And one kind of point I really want to bring your attention to is that for Drosophila prolongata, we're seeing, again, this strong positive correlation, which is not what we would expect under the gene capture model. Because the species exhibits male bias size dimorphism, we would expect, again, this strong negative correlation. But again, it looks like the rest of the phylogeny, which is weird. And then another weird point that I want to warn you is probably not that weird, is you're seeing this really strong negative correlation in Fuyamai, but this species just didn't really react well under our low conditions, so we have very small sample sizes. And so we have really small sample sizes, and so the relationship might not be as true to what you're seeing. But generally, our results are not very consistent with what we would expect under the gene capture model. And so when I zoom in on Drosophila prolongata, and remember the species is showing this classic male-biased, exaggerated trait, we see that as the traits become increasingly male-biased dimorphic, which would be near the -2, and in this case, our most sexually dimorphic trait is actually tibia width, we see that this trait is actually the least condition dependent, which is, again, not what we expect under the gene capture model. So to summarize, our results are not really consistent with the expectations under the gene capture model. And when we look across the phylogeny, we're seeing this very weak correlation between the evolution of dimorphism and condition dependence. And in that one species, Drosophila prolongata, that exhibits this kind of classic male-biased exaggerated trait, not only are we not seeing the expected relationship between size dimorphism and condition dependence, but we're seeing the opposite of what we would expect. So there's clearly something really interesting going on in Drosophila prolongata, and so I've just finished an experiment where I've repeated this condition manipulation in prolongata and closely related species to try to fill in that gap and maybe try to get an idea of what and how this trait is evolving. And then we're also looking to do some genomic work looking at the patterns of sex-biased gene expression in our high and low condition treatment for the Drosophila prolongata and closely related species in the row below clade. And we're looking to kind of refine our models. And thank you for listening. I'm really open to any suggestions. Thank you. I don't know.